For decades, the biological understanding of aging has been anchored to the Hayflick limit. This principle dictates that somatic cells possess a finite replicative capacity, eventually grinding to a complete, rigid halt. This lifespan is dictated by the progressive burning down of telomeres. Telomeres are the protective ribonucleoprotein caps located at the physical ends of linear chromosomes. They are strictly maintained only in specific cell populations where endogenous telomerase, an enzyme that builds telomere length, remains active. Once an individual cell's telomeres reach a critical minimum length, the chromosomal ends become exposed. This triggers a DNA damage response that forces the cell into terminal replicative senescence, an irreversible state where the cell permanently stops dividing. These senescent cells do not simply die. They begin to secrete a toxic pro-inflammatory mixture of cytokines and proteases known as the senescence-associated secretory phenotype, or SASP. The localized release of SASP actively disrupts the surrounding tissue homeostasis. It spreads cellular stress to neighboring healthy cells, accelerating systemic physical decline across the entire organism. Under this orthodox framework of molecular biology, organismal aging is understood as an inevitable autonomous failure. It is a process isolated within the nucleus of individual cells. Recent investigations, spearheaded by researchers from Centcell and Oxford University, have detailed a completely telomerase-independent mechanism of rejuvenation. They identified distinct, highly structured extracellular lipid vesicles. Because they organize into defined, flowing networks within biological fluids, these structures have been named telomere rivers. These specific vesicles carry a highly complex molecular cargo, anchored by fragments of cleaved telomeric DNA. The adaptive immune system actively generates, packages, and distributes these rivers throughout the bloodstream. Once in circulation, they migrate to distantly located senescent tissues across the body. Upon arrival, they integrate their genetic material to reverse established biochemical markers of aging. Telomere maintenance is a highly coordinated, immune-driven systemic network capable of broadcasting biological youth from metabolically healthy cells to failing tissues. The genesis of these rivers requires a specific catalyst. It is strictly an antigen-driven response initiated during an active immune challenge. The entire process begins with the formation of an immunological synapse between an antigen-presenting cell, or APC, and a recipient CD4-positive T lymphocyte. An APC is a specialized immune cell that processes and presents foreign antigens to T cells to trigger a response. Forming this stable synapse directly triggers a massive, rapid influx of intracellular calcium ions within the donor APC. This specific calcium cascade forces the targeted degradation of the shelterin complex. Shelterin is a specialized 6-protein structure that protects the physical ends of linear chromosomes. Its destruction leaves the APC's own chromosomes biochemically uncapped. An enzyme called TZAP is immediately recruited to the exposed ends. TZAP is a regulatory protein that enzymatically cleaves terminal telomeric DNA to maintain chromosomal homeostasis. Here, it physically shears off double-stranded fragments of the TTTAGGG sequence from the APC. The newly liberated telomeric DNA is routed to the cellular periphery and packaged into a discrete extracellular vesicle alongside RAD51. RAD51 is an enzyme essential for homologous recombination, the process of exchanging genetic material between similar DNA molecules. The inclusion of RAD51 is an evolutionary necessity. Without this specific enzymatic machinery, the donated telomeres would simply degrade rather than successfully fuse with the chromosomes of recipient cells. To create the raw biological material required for systemic tissue maintenance, the innate immune system literally sacrifices its own chromosomal caps. Following intracellular packaging, the APC actively transports these telomere-laden precursor vesicles across the microscopic gap of the synapse and directly into the cytoplasm of the recipient CD4-positive T cell. In the immediate aftermath, the recipient T cell utilizes a significant portion of this donated DNA to enzymatically elongate its own chromosomes. This buffers the lymphocyte against activation-induced replicative exhaustion. The T cell does not keep all of the acquired genetic material. It actively recycles approximately 40 to 50% of the acquired telomeric vesicles, preparing them for systemic redistribution. Releasing raw precursor vesicles carries significant risks. They are loaded with GAPDH, a key enzyme that aggressively drives glycolysis. This metabolic state in recipient tissues is associated with cellular senescence and malignant transformation. To solve this, the recipient T cell undergoes asymmetric division, a biological process where a single parent cell divides into two distinctly different daughter cells. It achieves this by unequally segregating its internal components. During this specific mitotic division, the emerging stem-like T cells physically compartmentalize the surplus telomere vesicles, segregating them away from their highly active glycolytic machinery. This comparative chart shows the resulting proteomic divergence. The precursor vesicle on the left is packed with GAPDH. The mature telomere river on the right shows a targeted depletion of GAPDH, replaced by specific stemness factors like Notch1 and beta-catenin. The deliberate enrichment with these developmental transcription proteins establishes the vesicle as a targeted tissue reprogramming agent. T cells act as a biological processing hub, meticulously curating the genetic payload to deliver vitality without triggering malignant transformation. The structural assembly, the asymmetric segregation, and their ultimate systemic release all require active mitochondrial fatty acid oxidation, or FAO. FAO is the biochemical process of breaking down lipid molecules to generate adenosine triphosphate, or ATP. The biochemical gatekeeper of this entire pathway is carnitine palmitoyltransferase 1A, or CPT1A. CPT1A is the rate-limiting enzyme that conjugates long-chain fatty acids to carnitine, allowing them to enter the mitochondria. In vivo, histological analyses of reactive lymph nodes prove that extracellular telomeric material is found almost exclusively decorating memory T cells that express high levels of CPT1A. Fatty acid oxidation tightly regulates the cellular lipidome by upregulating ceramidase. Ceramidase is an enzyme that aggressively breaks down stress-inducing lipids. This metabolic flux forcibly blocks de novo synthesis and accumulation of ceramides, which are rigid, bioactive lipids that cause severe cellular stress. Concurrently, this pathway generates high levels of phosphatidylethylamine, or PE. Phosphatidylethylamine is a primary phospholipid that increases membrane fluidity and stabilizes protein complexes. Without a stable, fluid synapse architecture, driven by this exact lipid composition, the massive intercellular transfer of genetic material cannot be sustained. The physical capability of an organism to package and transfer youth-promoting DNA is strictly gated by the underlying lipidomic health and mitochondrial efficiency of its T cells. The aging process in mammals is universally characterized by inflammaging. This is a state of chronic, low-grade systemic inflammation that permeates the entire body. This persistent inflammatory pressure forcefully shifts the metabolic profile of immune cells. It pushes them away from efficient mitochondrial fatty acid oxidation and toward aerobic glycolysis. When researchers examined senescent T cells from older demographics, they found that these aged lymphocytes almost entirely lack CPT1A expression. Deprived of the ability to oxidize fatty acids, these cells divert their lipid resources into the catastrophic accumulation of toxic ceramides. The resulting membrane rigidity impairs T cell receptor signaling and blocks their ability to acquire telomeric vesicles from APCs. Without the initial acquisition, the endogenous production and systemic release of telomere rivers ceases completely. The somatic tissues that most desperately need systemic rejuvenation eventually lose the highly specific metabolic machinery required to produce it. Once processed and released by healthy T cells, mature telomere rivers navigate the vascular networks and extravasate into the extracellular matrix of distant organs. Here, they exhibit a remarkable tropism, specifically targeting and penetrating terminally differentiated senescent cells. Upon assimilation, the retained Rad51 recombination factor mediates the seamless physical fusion of the donated 3,000 base pair telomeric DNA fragments directly onto the critically shortened chromosomes of the host cell. This mechanism entirely bypasses the biological requirement for endogenous telomerase expression. It provides a safe, exogenous source of chromosomal extension without requiring dangerous alterations to the host's gene expression. Simultaneously, the payload of enriched stemness factors drives profound cellular reprogramming. Exhaustive assessments showed this reverses the enzymatic activity of senescence-associated beta-galactositis, or beta-gal. Beta-gal is a lysosomal enzyme used as a primary biomarker for cells that have ceased dividing. This line graph tracking survival curves shows the findings from the Sencell and Oxford in vivo trials. When purified livers were administered to an aging cohort of 20-month-old mice, the median lifespan increased by 17 months. In the context of standard murine biology, a 17-month extension equates to 54 human years. Several treated mice survived to nearly 5 years of absolute age, shattering the standard maximum lifespan ceiling of the species. Providing a safe, exogenous method of telomeric extension actively breaks established mammalian biological limits. The conformation of telomere rivers resolves the decades-old enigma of heterochronic parabiosis. This is the experimental surgical joining of the circulatory systems of a young and an old organism. Rivers prove that actively circulating DNA and stemness factors, deeply tied to systemic metabolic health, act as the exact rejuvenating engine. The exact same GAP-DHA-negative telomeric vesicles have been successfully isolated from the rudimentary xylem sap of plants, specifically terrestrial orchids. In flora, empirical data demonstrates that the formation of these rivers strictly requires photosynthesis-driven beta-oxidation, perfectly mirroring the mammalian immune system's reliance on mitochondrial fatty acid oxidation. Long before the evolution of specialized circulatory and adaptive immune systems, primitive multicellular entities required a reliable metabolic method to redistribute genetic vitality from metabolically healthy peripheral cells to deeply localized internal tissues. The presence of this shared metabolic dependency implies that telomere rivers are the direct biological manifestation of a primordial cross-kingdom program of genetic altruism. The isolation of these structures immediately suggests therapeutic applications. Translational researchers have already begun engineering synthetic variants, known as artificial rivers, to bypass the need for natural immune generation. By utilizing RNA interference, scientists have artificially silenced the GAP-ADH gene in donor cells to generate highly potent, purified vesicles ex vivo. RNA interference is a biological process where double-stranded RNA molecules inhibit gene expression by neutralizing specific mRNA molecules. An alternative, completely pharmacological approach involves using targeted CPT1A activators to force the in vivo metabolic reprogramming of aged T cells, reigniting the patient's own endogenous river production. The aggressive pace of commercial and academic development indicates that Phase 1 human clinical trials, aimed at evaluating the foundational safety of these river-based therapies, are targeted to commence by 2026. The discovery of telomere through the manipulation of systemic genetic transfer.